Genes are like the story, and DNA is the language that a story is written in. And the greatest history book ever written is hidden in our DNAs. Why I'm talking about DNAs today. Good morning, everyone. Myself, Dr. Sakshi Bhardwaj, on behalf of Phantom Technologies, I would like to welcome you all and much obliged to have you all here. In this subsequent Women's Day special webinar series session, which is entitled as Targeting DNA Methylation in Head and Neck Cancer, an Insilco study. We are fortunate to have such a special guest speaker with us today who has expertise in omics. It gives me immense pleasure to introduce our speaker of the day, Dr. Zainab Siddiqui, Assistant Professor at the Center for Disease Mapping and Therapeutics Research, and she is an in charge research metabolic unit head from Agri-University, Lucknow. She has done her PhD in biotechnology from Lucknow, Uttar Pradesh, her master's and undergraduation from Jamia University, Delhi. She has received many awards and grants to her credentials. She is a member of the Indian Science Congress Association and European Society for Medical Oncology, Lugano, Tiziano. She is also on the editorial board for many respected journals and a member of Scientific Development Committee for investigating anti-cancer property of natural compounds at the Asian Pacific Journal of Cancer Prevention. Her credential also includes two book chapters and more than 14 publications. Dr. Siddiqui's discussion today will focus on development of in silico approaches to target DNA methylation in neck and head cancers. Now, without taking much time, I would like to hand over the platform to our guest of speaker. Over to you, ma'am.
Thank you, Dr. Sakshi, for the introduction. A very good morning to all of you. Before beginning with my talk, I would like to thank the organizers for inviting me to this Women's Week program. I'm very much obliged to them. This year's International Women's Day theme is hashtag Inspire Inclusion to celebrate women's achievement, raise awareness about discrimination, and to take action to drive gender parity. IWD belongs to everyone, everywhere. Inclusion means all IWD action is valid. Now, today's talk is on targeting DNA methylation in head and neck cancer and in silico study. There will be two parts of this discussion. One is the DNA methylation, and the other one is the head and neck cancer. 33% of men and 18% of women use smokeless tobacco in India. It accounts for 27.5 crore people in India, and Tata Memorial Hospital and the Sheikh Saria Institute of Public Health has taken an initiative to run the anti-tobacco mass media campaign featuring Mukesh and Sunita Tomar. Both of them died at a very young age despite cancer treatment. Hence, this cancer is of major concern. Now, the Globocan is an initiative of WHO, and it is maintained by International Agency for Cancer Research. And Globocan 2020 predicts the incidence rate by 2040 to be 47.1% in men and 46.9% in women. The mortality rate will also see a increase by 2040. That is, in males it will be 50.8%, and in females it will be 53%. Overall, this cancer ranks 7th globally, and in India the incidence rate to male versus female is 0.4 is to 1. Now, as I was going through the cancer statistics, what we found was that Asia covers the largest area, that is 63.9%. And further looking into it, we found that India and its neighboring countries like Bangladesh and Sri Lanka and Pakistan, they are majorly affected by this disease. Furthermore, the second part of our talk is DNA methylation. So for long, cancer scientists are working on the cancer genetics, but from the last 2 decades, they have moved their focus to the cancer epigenetics. Now, DNA methylation is a part of epigenetics and it is affected by several external factors. These are, in case of head and neck cancer, these factors can be tobacco abuse or your betel quid chewing and alcohol intake. DNA methylation also plays a role in gene regulation, and this process is catalyzed by an enzyme known as DNA methyltransferase. And there are several subtypes to it, like DNMT1, 2, 3A, 3B, and 3L. It has been seen that 60% of the mammalian promoters have the CpG islands. These are the sites where your methyl groups are being added. As you can see here in the pictorial representation, that the nitrogenous base cytosine is being methylated by the DNMT at the fifth carbon position, and the methyl donor is S-adenosyl L-methionine, which is known by the abbreviation of SAM. Next, it is the best epigenetic regulators apart from histone modifications. These are also the favorite sites of methylation and the known coding RNAs. Now, why this DNA methylation is important for us? First of all, it helps in the identification of newly synthesized DNA strands. Here you can see the parental DNA strand, which is being fully methylated. And after replication, what happens is that the newly synthesized strands are devoid of methylation. So one can easily identify the newly synthesized strands from the existing methylated parental ones. The second most important factor is that DNA methylation regulates the gene expression, and it has been observed that it is involved in gene silencing. As I told you earlier, that promoter regions are methylated by DNMTs. So hypermethylation of the promoter region switches off the transcription of genes. And these genes can be your tumor suppressor genes like P16, P15, and many more. And these can also be DNA repair enzymes genes or metabolism of xenobiotics. For long, it was stated that hypermethylation leads to gene silencing, but later on in the year 2002, a study was conducted by Gouliard et al., and he was the first one to identify that hypermethylation leads to the gene activation. He was working on the enzyme activity of telomerase gene, and this gene was found to be highly expressed in cancer models upon hypermethylation. So you can say that the genes whose transcription is being switched on are basically the oncogenes. So these can be BIRC5, FOXA2, PAX2, and many more. Furthermore, this is a kind of a pathway representation. It was taken from a database Reactome. It is a pathway database. At the center of the diagram, you can see the DNMT1 gene marker, and it is regulating 22 other gene expression. So this, this signifies that how it regulates other genes expression also. In the right hand side is a tabular representation where all the DNMT subtypes functions have been mentioned. DNMT1 and 2, they both are responsible for DNA methylation, but DNMT1 has an additional factor, that is the maintenance. And by maintenance, we mean that the hemimethylated DNA strands are fully methylated by DNMT1, while DNMT3A and 3B, they carry out the de novo methylation. So de novo methylation means all the unmethylated DNAs are characterized by these DNMTs, while DNMT3L works synergistically in conjunction with 3A and 3B. It doesn't work alone, and it enhances the activity of the other two DNMTs. Now, coming to the section where we have employed the methods and what were the outcomes of the methodology we employed in this study to characterize the functions of these DNMTs, we'll be discussing this. So first of all, as my topic is on protein docking, so to dock a protein, we need a protein and a ligand. So the protein targets were identified, and its significance I have already stated that these are the DNMTs and how important is to study them in head and neck cancer. Now coming to the ligand part, the ligand selected for our study were the phytochemicals. So this phytochemical library was prepared through a literature search survey, and there were several keywords that were used to obtain the research articles that signify the importance of phytochemicals in the regulation of DNMTs. And these articles were selected from MASH, and they were the keywords used were the DNA methyltransferase, cancer, dietary phytochemical, natural active compounds, and DNMTs. Furthermore, few inclusion and exclusion criteria were laid down to filter the articles. So the articles that were showing significant association of the phytochemical with the DNMT were selected, and we have made an effort that the articles which were mentioning this significant effect should be carried on or tested on cancer animal models or cancer cell lines. Furthermore, the studies that were using in silico methods or review articles, or the studies that were available on active compounds isolated from non-plant sources such as your microbes and fungal and sponges, were excluded from the study. After performing this criteria, inclusion and exclusion criteria, we finally led to the inclusion of 41 articles, and in these 41 articles approximately 33 phytochemicals were mentioned. So this is a tabular view of the 33 phytochemicals that were obtained from the 41 research articles, and the articles were further characterized what type of cancer cell lines they were using, and what were the methods they used to detect the activity of the phytochemical on the target protein. So when we were analyzing these, we found that majority of the articles, that was like 90.37%, were done on cancer cell line models. Furthermore, looking into it, we got to know that majority of the diseases that were taken up were on breast cancer, followed up by prostate cancer and colorectal cancer. So to perform protein docking, one needs to know about the 3D structures of both the ligand and the protein. So the ligand structures can be downloaded from PubChem, ChemSpider, or ChEMBL. There are several online tools available. While the 3D structures of target proteins can be downloaded from PDB or UniProt. As the DNMT2 proteins structure was available on the PDB, but the remaining 4 DNMT structures was not available. So we needed to create or predict a 3D model for these remaining 3 structures. For this, we use the technique known as homology modeling. So this technique is basically based on the unknown sequence, that is the DNMT of DNMT1, 3A, 3B, and 3L, are used as target sequences, and for them a template sequence is being searched, and the similarity between these target and template is done through BLAST-P. So the BLAST-P gives you a sequence identity or sequence similarity, how homologous is your template with the target sequence. So basically it should be greater than 33%. Few articles mention that it should be greater than 35% or 40%. So once you match them, you have to select the best template, because these softwares provide you a list of template sequences and you have to select the best one from it. So for selecting the best one, you need to consider the few parameters like the global model quality estimate or the Q mean disco global score. So the GMQE is actually the quality estimate which combines properties from the target template alignment and the template structure. It predicts the LDT score of the resulting model. So what is this LDT? LDT is basically a local distance difference test. So it measures the distance between the carbon alpha atoms or the backbone atoms. So it is helpful in selecting the optimal templates for modeling. So it ranges between 0 to 1. The more it is closer to 1, then it is considered a good model. In our case, we got good scores for DNMT1 and 3L, while 3 and 3B didn't show that much of good score for GMQE, but the rest parameters were satisfied by 3 and 3B. The next parameter is Cumin Disco Global Score. So it is an error estimate. So it employs statistical potentials of mean force and agreement terms by introducing a consensus based distance constraint. This DISCO exploits distance distribution from experimentally determined protein structures that are homologous to the model being assessed. So the template selected for DNMT1 was 4WXX, and for 3A and 3B was 6PA7, while for 3L was 8EIK. Now, next we wanted to validate the 3D models that were generated by using online softwares. So for this validation, we performed the Ramachandran plot analysis. So Ramachandran plot analysis basically tells us about the protein amino acid, the amino acid residues which are present in protein, their conformations is in the allowed region or not. The more your structure is belongs to the allowed region, then it is said to be a good protein and it can be validated. So the red regions in the diagram, as you can see in the Ramachandran plot, these are the allowed regions, while the yellow one indicates that these conformations are permissible if a little flexibility is allowed in the bond angles, while the remaining white region, or you can say the unshaded portion, it indicates sterically disallowed conformations. Now coming back to the previous slides, here you can see the amino acid residues of these four proteins basically lied in the regions of 88% to 90%, and these were the favored regions, the red ones and the yellow ones. While those in the disallowed region, we didn't find any amino acid residue in the white portion or the unshaded portion. So we can say that the modeled proteins are good to go and then can be considered further for virtual screening or protein docking. So before beginning about protein docking, we need to take some reference compounds, or you can say that they can serve as controls. You need to know a reference compound or a control before comparing your results with the phytochemicals docked targets. So these reference compounds were the FDA approved DNMTs inhibitors, and these were decitabine, azacitidine, cytidine, and zebularine. So when we docked these compounds with all the DNMTs, 5 DNMT enzymes, we found decitabine to showing the lowest binding energy and a strong affinity to our target proteins. In these circles, you can see the 3-letter code of amino acid and the position is being mentioned. And the dotted colored lines shows the type of interaction or a type of bond it is forming with the target proteins. And these amino acids, these are the active site residues of your target proteins. And this chemical structure is your decitabine. So only decitabine showed significant results with DNMT1, and this was the only epi drug, or you can say the only approved FDA drug which we dogged and found to be showing the least binding energies with all the remaining DNMTs. For DNMT2 also you can see it's showing the lowest binding energy of -10. Further, for 3A also, it is showing -15. It is the lowest and it depicts the strong binding affinity to the target protein. Furthermore, for 3B also, it is showing the same, the lowest binding energy, that is -10.6. And then for 3L also, it is showing the lowest one, like -10.6. So once we were done with this, then we moved to the docking, performing docking of our ligands, 33 ligands that we formulated in our phytochemical library, and we docked it against the respective target proteins. And the top three compounds showed a low binding energies were rosmarinic acid. It showed a docking energy of -49.4. It is the C docker energy. For epigallocatechin gallate or EGCG, it was -48.5. and for procyanidin B2, it was minus 37.3. In case of rosmarinic acid, you can see the amino active site amino acid residues were cysteine, proline, arginine, glutamate, valine, and aspartate. While EGCG also comprise these amino acid apart from others, like your leucine was over there, and same with procyanidin B2. It showed valine, glutamate, and arginine in the active site. Then next we did protein docking with a second target, that is DNMT2. And the top three compounds were epigallocatechin, procyanidin B2, and rosmarinic acid. The docking C Docker energy or the docking score for epigallocatechin was minus 55, procyanidin B2 was minus 51, followed by rosmarinic acid, that is minus 46.8. Now comes the third protein, that is DNMT3A. When we docked it against the ligand library, the top compound which shows the highest affinity, strong affinity, or the lowest C Docker energy was minus 54.9. Next one was the epigallocatechin gallate. It was minus 54.6, and quercetin, which was minus 42. Furthermore, DNMT3B, it showed the top three compounds that depicted the least binding energies were corosolic acid, epigallocatechin gallate, and rosmarinic acid. Corosolic acid showed the least binding energy amongst all the phytochemicals that were studied, that was minus 75.2. Now next one is the DNMT3L. The top three compounds that were retrieved from protein docking were epigallocatechin gallate, which had a C Docker energy of minus 50, and rosmarinic acid had an energy of minus 48, and silibinin had an energy of minus 41.2. Now, once we have selected these compounds, we further moved on to estimate the properties, or you can say that drug likeness, how they can behave, how well they can behave as a drug. So, for this, several rules have been put forward by several scientists. The most acceptable one or the most famous one is the Lipinski's rule of five. So for Lipinski's rule of five validation, the molecular weight of the compound that you are proposing as a drug should be less than 500 Dalton. And the log P value should also be less than 5. And hydrogen bond donor should be less than 5. And your hydrogen bond donors are OH groups or the NH groups ones. Then hydrogen bond acceptors should be less than 10. These are the nitrogen and the oxygen molecules. So there is Estek's rule of 3 also, and there is 3 by 75 rule and a bioavailability score. Then there is a new in topic that's being introduced, which is the beyond Lipinski's rule of 5. So at the time when this Lipinski's rule of 5 was introduced, there were the articles that were utilizing it were not available online at that time. So the proper validation or the shortcomings were not put forward to the scientists or the researchers who were working on it. So in the year 2019, Schulz published an article, and he included FDA approved oral new chemical entities from 2008 to 2017, and it comprised 214 drugs. So in the top row of this article shows the 1997 90th percentile. This is the Lipinski's group or the rule following drugs. And next one below this is a row in which Schulz published the data related to the drugs that were available from 2008 to 2017. So here you can see the drugs that were introduced in the Lipinski's rule of 5. They had a molecular weight ranging to 470 as an average. But by Schulz, it was predicted that the molecular weight of the compounds that he included was 607, which is more than 500 delta, and it was significantly statistically different. Then the hydrogen bond donors were similar to the Lipinski's rule. Schultz found no change in it. Then there was hydrogen bond acceptors. Schultz found increased hydrogen bond acceptors count, that is 11.7. Then there was the TPSA, that is the topological polar surface area, which was not significantly different from the Lipinski's rule of 5 in the Schultz study. Then there was ROTB, that is rotatable bonds, and these were significantly higher in the Schultz study. Then there was FsP3, that is fraction of sp3 carbon atoms. It was found to be significantly reduced in the Schultz study. Now there was another parameter that was considered by Schultz, that was aromatic number of aromatic rings present in the ligands which are being proposed as drugs. So this count was also 4, and it was significantly higher than of the drugs that were used in the Lipinski's rule of 5. Furthermore, there was another study published by Bennett, and he authenticated that earlier, when this rule, Lipinski's rule of 5, was put forward, the drugs transporters data was not available. But now we are in the era where we know the significance of the transporters and how the drug influx and efflux is being associated with their activity. So what they said was that the drugs are substrates for some transporter, and its information is not included in the rule of 5. So it should be included and considered. So further, we characterize the drug likeness properties of our compounds. These were rosmarinic acid, EGCG, procyanidin B2, quercetin, porcelic acid, and silibinin. So the molecular weight was less than 500, but only for procyanidin B2, we observed the molecular weight to be 578, which is greater than 500. But as we go according to the beyond the rule of 5, then it can be considered or proposed as a drug. Now coming to the water solubility, the log values that we got was good enough to identify its solubility. Then there was log P. It was similarly was good enough to propose them as drugs. Then for the hydrogen bond donors, the count should be less than 5, but for EGCG and procyanidin B2, it was greater than 5, 8 and 10. So this rule was being violated by these two compounds. Then comes the hydrogen bond acceptor. So for hydrogen bond acceptors, the count can be more than 10, but for as put forward by Schultz, that it can be greater than 10, like 11. So only procyanidin B2 showed 12 hydrogen bond acceptor count. So this rule was also violated by procyanidin B2. Then there is rotatable bond. So the rotatable bonds count was also good enough to propose them as drugs. So it's been said that if your compounds which you are proposing are violating one or two rule, then it can be considered as drugs. They cannot be excluded from the list. So we considered them and moved on forward with it. So there is a drug likeness model score plot was also plotted to find out if the compounds were showing positive scores, and the ones with the positive scores behave as drugs. For silibinin, we got a score of 0.8. For procyanidin B2, we got a drug score of 0.7. For EGCG, it was 0.2. It was positive. And for corosolic acid, it was 0.6. And for rosmarinic, it was 0.3, followed by quercetin, which was 0.5. So all of these showed a positive scores and can be considered as drugs or proposed as epidrugs. The drugs which regulate the epigenetics are termed as epidrugs. So next we perform the ADMET analysis. So ADMET stands for absorption, digestion, metabolism, excretion, and toxicity. So this was done through the DS client server system 2022, which is being employed in our university. So the compounds were tested for these properties. So the first parameter is the permeability in the blood-brain barrier. So the compound which we selected were for head and neck cancer, and these were not rating any brain disease. So if they are showing a higher score, that is 4, then we can consider them, because these are going for the treatment of head and neck cancer and not for the brain tumors or any brain disease. So their probability is not an issue that they need to cross the blood brain barrier to be effective. So that gut metabolism is important, which is to be considered. And for gut metabolism, you need to know the rule of the cytochrome P450s. The cytochrome P450s are the group of metabolizing enzymes, and they are present in the liver where they carry out the metabolism. Then there are several subtypes to it. The subtypes which DS being considered is CYP 2D6. So the CYP 2D6 values were 0.01 for rosmarinic acid. So when it is closer to 0, then you can see it's a non-inhibitor. And for EGCG, it was exactly 0. For prostaglandin B2 also it was 0. Like these were non-inhibitors of the drug metabolizing enzyme, so it can be metabolized in your system. Then there is quercetin. It is also closer to like 0.1. And then for corosolic acid it was 0.04, and silibinin was 0. Next, we want to know if these can be hepatotoxins or they can behave as toxins in our liver system. So for to be hepatotoxic, the value of 0 tells us that it is a non-toxic substance. If it's 1, then you can say it is a toxic substance. So all these compounds had values less than 1. Like quercetin had 0.9, which was quite closer to 1, but still it was less than 1. Then for the solubility also, the solubility of these compounds ranged between 3 to 1. So the solubility is basically the human intestinal absorption level. 0 stands for good absorption, 1 for moderate, and 2 for low absorption, and 3 for very low absorption. So there is another parameter, that is ppb. It is the plasma protein binding. So there are several plasma proteins available in our blood systems or circulatory system which binds to protein, and then they carry out the function. They take them to their target organ or the target regions where they are required. So for traveling of proteins in the circulatory system, you require plasma proteins, and their binding affinity should be checked on, so that you can know that your compound is being transported in the system. So the plasma protein binding acceptable range is from 0 to 2. So as you can see, all the compounds, phytochemicals, had the range lying between 0 to 2. Rosmarinic acid had the value 0, EGCG had the value 0, prostaglandin B2 was also 0, and quercetin was 0.61, corosolic acid was 1, and silibinin was 0. Next is the mutagenicity. So, is it mutagenic or not? This is being tested, and it's been predicted through MES test by the ADMET analysis. So, only quercetin was found to be mutagenic, while rest were non-mutagenic. So, these compounds can be said that they are good to go. Now, coming to the last section, that is conclusion of our data. First one is that the compounds that were identified for head and neck cancer therapy, these were EGCG, rosmarinic acid, and prostaglandin B2 for DNMT 1 and 2. While for DNMT 3A, we had EGCG, rosmarinic acid, and quercetin. For DNMT 3B, it was EGCG, rosmarinic acid, and corosolic acid. And DNMT 3L was EGCG, rosmarinic acid, and silibin. So what was common in all the 4 DNMTs was that EGCG as rosmarinic acid were the top 2 compounds and common to all the DNMTs. So they can be proposed as potent epi-drugs or potent inhibitors of DNMTs in the long run if they are validated, or molecular simulation is being further carried out, because we didn't completed this study as a whole. So further actions that are being proposed is the molecular simulation of this compound to finalize or pinpoint the exact ones which are going to be considered as potent inhibitors of DNMTs. So this study also reveals novel targets for interaction with HNC biomarkers, and they can be used in research as anti-cancer therapy development. And docking and admit screening confirmed the strong binding affinity and drug-like properties of these phytochemicals, respectively. Thank you, everyone. That's the end of my topic. And I'm over to you, Dr. Sakshi. I'm free to answer any questions.
Yeah, thank you so much, ma'am. It was a very informative session. We have some questions. Let us take
Yes.
these questions.
Okay.
Yes, the first question is, can you give us an idea how to find a reproposable for selected target?
Selected targets. Okay.
Yes.
So if you want to select a target, there are several databases also available. There is a biomarker. It depends on the type of interaction you are going to study. For in my case, I wanted to study DNA methylation. So DNA methylation is carried out by DNMTs only, and methylation itself has a significant role in the pathogenesis of head and neck cancer. The genes which it is regulating are the tumor suppressor genes and the oncogenes. If you want to look into the another aspects, suppose you want to know about the microRNAs, so there is a database known as miRNA. You can identify the microRNAs, non-coding RNAs. There are several databases, and usually people go by the literature based approach for identifying the targets. First, you have to identify the model which you are going to study, the cancer model or any disease model. It could be like diabetes or anything. Then you have to pinpoint the area of interest that you want to look forward in it. Suppose if you are taking studying about cancer, then you are more interested in knowing about the proliferation or the apoptosis of the cancer cells. Then once you are going into it, then you have to identify key biomarkers that are regulating the outcome of the process, like in the case of cancer proliferation you are taking. So you have to identify the key biomarkers and the signaling cascades. First, you have to pinpoint the oncogenes that are affecting it and the cascades that are regulating it like RAS-RAF or the PI3K-AKT, and your oncogenes can be like they are many more. They can be C-June, MYCs, they are your oncogenes. These can be proposed as targets. And even can also go the other way around also. You can see the current cancer drugs which are being proposed, what are their molecular targets and how ineffective it is, or what are the limitations with it. So you can improvise it. You can look into the limitations that it has, and you can improvise your study by looking further into it.
Yes. Next question. Why only specific DNMT were selected for docking studies?
Because methylation epigenetics plays an important role in cancer pathogenesis. As I told you that the 60% of the mammalian promoter regions have these CPG islands. So these CPG islands are the hub for DNA methylation. The cytosine residues, the GC content is mostly more than 50%. So these are the targets for DNMTs. So if you are having promoters like more than 50% that compromise the CPG island, so they are more prone to DNA methylation. And this epigenetic is affected by external factors. And the major risk factor in the case of head and neck cancer is the use of tobacco. And in Asia, particularly in India, there are several smokeless tobacco chewers or consumers in India. So this is the most aggressive form of cancer, and it is being affected by the external factor. Epigenetics is basically being regulated by these risk factors.
Okay. Next question. Yes, can you tell us the food source of phytochemical rosmarinic acid, quercetin, epicachin, gallate, which you used in your studies?
So for rosmarinic acid, this is rosemary. For quercetin, there are several other food sources like buckwheat and onion. For epicatechin gallate, there is the excellent food source which is green tea.
Okay. Um, Next question is why homology modeling is significant during the drug designing?
As you can say, it is significant for those proteins in which your structure is being not identified. So the structure prediction or identification for experimental use is done by NMR and X-ray crystallography. So these are a costly affair. So to the computational based approach is homology modeling, by which you can predict your structure.
Yes. Which docking tool was used for your studies?
It was Discovery Studio 2022 version.
Okay. Even with the Discovery Studio, we have a separate module that is for the homology modeling, where you can pull out. You have mentioned the Ramachandran plot itself, so even you can
Yes, yes.
analyze the Ramachandran plot there, where you can see. Yes, the importance of Ramachandran plot is also for to analyze whether is there any gaps in between the amino acid. Basically, we are going to use the compact structure of your macromolecule for your studies, like for the docking studies. So hence these are the little techniques which are helpful while during those docking studies and further simulation. Yes, next is okay. This is the same, I think, tailing question for this repurposing of the selected target protein actually for the oral squamous cell carcinoma. I think she was asking about this particular oral cell
Carcinoma.
carcinoma.
It,
Yes.
it is a subtype of head and neck cancer.
Yes. So, um, what is the effect of these phytochemicals on the normal cells?
On the normal cells, okay, this was basically on cancer cells. So for the normal cells, we have not assessed their role, and it could be good enough, it could regulate their function, because normal cells also undergo cell cycle phenomena. So the cell cycle phenomena is to be regulated. The more cell cycle you are having, it will lead to the formation of cancer or cancer proliferation. So it can keep a check on normal cells to not undergo an abrupt cyclings of the cells, which can lead to cancer. So it's like prevention is better than cure kind of thing.
Okay, so I think we have done with all the questions. So thank you so much, ma'am, for it was very
Thank you.
informative session. Um,
Thank you.
Now it's time to felicitate our speaker of the day with the memento and certification of appreciation. Thank you so much, ma'am, once again
Thank you.
for giving your time and for such a great presentation. It was pleasure to have you here with us today. And stay tuned, us guys. Let's connect and grow together. Thank you, thank you, thank you all once again
Thank you.
for joining us. Thank you.